introduction. I want to thank the committee for giving me the opportunity to present my data in this great symposium. It is a great way to get your feedback, so I really want to thank everyone, especially the ones in Israel who stayed so late. The brain is highly vascularized organ, but also extremely tightly regulated in order to maintain neuronal function. The endothelial cells lining the blood vessels, as shown here, they form the blood-brain barrier, or BBB in short. These cells have two unique properties that enable this barrier function. First, they have specialized tight junctions that prevent the diffusion of molecules between endothelial cells, and they have low rates of vesicular transport, which limits the delivery of solutes from the blood to the brain. As an example, if I inject a small molecule tracer to the bloodstream of mice and look at the brain, the tracer will be confined to the barrier-containing vessels, which show up as strings in this slice, and this is what we call leakage assay. If we repeat this assay in mutant mice, the tracer will leak into the parenchyma, as shown here. The blood-brain barrier is important for normal brain function, but from clinical point of view, it is a hurdle. A functional BBB is a major option, shown one in here. On the other hand, recent studies have shown that BBB breakdown is implicated in neurodegenerations in conditions like Alzheimer's. So there is a big interest in the field to be able to manipulate the barrier, namely to transiently open it to allow for drug delivery or to tighten the barrier, the compromised barrier in patients. But to do that, we must have a better understanding of the molecular mechanisms that govern the BBB, and these are still largely unknown. My research aimed to uncover as many regulators of the BBB as possible. And for that, I wanted to identify candidates first. I won't be showing that today, but I did RNA profiling comparing CNS endothelial cells to leaky endothelial cells and found about 200 genes that I call promising candidates. I then wanted to develop an in vivo system to test these candidates. And since I wanted to study as many genes as possible, I didn't want to be reliant on knockout mice, since this would be slow or impossible for some of those genes. We so far examined 16 genes and have 8 confirmed candidates. And naturally, the next step is to uncover the mechanism of action, but let's focus on our strategy first. I've developed a platform that takes advantage of the CRISPR-Cas9 system and a unique AAV isoform that can specifically infect brain endothelial cells, as shown here. This is the virus, and these are the blood vessels. Our viral construct candidates a guide-specific RNA, and these are injected into endothelial-specific Cas9 mice, and we get brain endothelial mutants within 3 weeks. To test this method, I've targeted a crucial BBB gene named GLUT1, which encodes the glucose transporter. And as you can see here in control animals, the virus does not interfere with GLUT1 expression that is widely expressed in blood vessels. But if we look at the knockdown animals, we see elimination of GLUT1 expression, specifically in those infected vessels, proving that we're able to silence target gene expression using our system. We then performed our leakage assay, and as you can see here, we saw leakage throughout the brain, confirming the expected outcome of the knockout mice. And motivated by this, we began our screen. And as I mentioned before, we observed leakage in 8 candidates, but today I will be focusing on 1 candidate. The gene is CCDC141. And just as my RNA-seq predicted, we can see that the mRNA shown in green is localized into endothelial cells. We can see that it is also enriched in the brain compared to a leaky lung vessels. In red, these are endothelial cells there. And when we look at the protein, we see that CCDC141 protein follows the same trend where brain endothelial cells have much higher levels of CCDC141 compared to lung endothelial cells. We then use our viral system to make CCDC141 knockdown animals and eliminate and examine those using the leakage assay as I shown before. As you can appreciate here, we saw severe leakage phenotype in CCDC141 knockdown animals. The tracer leaked out of the vasculature in multiple brain areas. And our next step, in order to dive deeper and understand what barrier properties those CCDC141 regulate, was to repeat these tracer injections, but with a unique tracer that can be visualized under electron microscopy and give us a closer look at the BBB. As I mentioned earlier, brain endothelial cells have two unique features. Let's focus on tight junctions first. These limit the diffusions of molecules between endothelial cells. Now, in this image, this is the lumen, this is the endothelial cell layer, and this is the brain. And a functional tight junction successfully halts the tracer, as shown here in the control animals, and here in CCDC141 knockdown animal, suggesting all of them have functional tight junctions. However, when we focus on the cytoplasm of these endothelial cells, we can see many tracer containing vesicles emerge in CCDC141 targeted mice. And if we look back at the control image, the cytoplasm would rarely contain transcytotic vesicles. At this stage, what caught my attention was the unique shape of these vesicles. Surprisingly, they were elongated tubular vesicles and not round as previously described in certain mutant mice. I quantified it and came up with this graph showing that in wild-type conditions, we would rarely see those vesicles, and in CCDC141 mice, they are becoming very abundant. Now, let me explain to you why I was excited about these results. Our lab has previously identified mutant mice that show elevated transcytosis of round vesicles made of caveolin-1. There's much effort to hijack this pathway to enable drug delivery to the brain. And we believe we uncovered a potential new pathway centering around tubular vesicles that is important for BBB functionality and does not depend on caveolin-1. I'm going to show you one piece of data supporting that hypothesis using an established protocol for caveolin-1 immuno-EM. We used gold conjugated antibodies to label CAV1, and we observed two things. One, there is no difference in CAV1 immunolabeling between the two samples. And second, the tubular vesicles that emerge in CCDC141 knockdown mice were not labeled by the antibody, suggesting they have a different molecular makeup. So what is the molecular makeup of these vesicles? We searched the literature for proteins that can induce membrane tubulation and eventually found paxin-2. I'm showing you a high magnification image of endothelial cells, and as you can appreciate here in the control mice, paxin-2 is not specifically enriched in a given cellular population in the brain, but when we look at the CCDC141 knockdown animals, we see increase in paxin-2 protein, specifically in brain endothelial cells. This was our first link between CCDC141 and an effector protein that may comprise these tubular vesicles. Now, our model is that CCDC141 is a new regulator of the BBB. It is inhibiting paxin-2 expression and prevent tubular vesicle formation. If we knock down CCDC141, we see increase in tubular vesicle formation and subsequent leakage. This model makes two testable hypotheses. First, that overexpression of paxin-2 would be sufficient to induce barrier breakdown. And second, that paxin-2 is required for leakage in CCDC141 knockdown animals. We tested both these hypotheses and both hold up. And here I'm showing you the data supporting the first. We generated paxin-2 overexpressing vectors and injected them to wild-type mice. The results were in line with our current model and show that paxin-2 is sufficient to induce leakage. Now, just for a quick summary, we believe that brain endothelial cells must actively inhibit multiple transcytotic pathways in order to maintain BBB function. These are caveolin-1 that was previously described. Today I discussed about CCDC and paxin-2, and we have other hits that we are currently studying. We're certain that the characterization of these molecular pathways will enable us to manipulate the barrier, namely to allow for drug delivery by transiently activating transcytosis, or to repair the compromised barrier by reactivating barrier-promoting genes in patients. More generally, I believe that within the next few years, our validation pipeline will uncover more core BBB regulators and will improve our understanding of the blood-brain barrier. I would like to thank my mentor, Chenghua, for being an excellent mentor and a great person to work with. Lucy Gordon, who joined the work and now spearhead the PAXIN2 project in the lab, and Joe and Jin Yu that worked to characterize my other confirmed candidates. And thank you for listening and being a great audience.